Once proteins are broken down to their individual amino acid constituents, what happens to the amino acids next, or what is the fate of amino acids once proteins are actually degraded? So, good day to all. Today, we are going to present to you the group 2. We are going to present to you the degradation of amino acids. As we start off, what are amino acids? You can read at the top. Amino acids are molecules that combine to form proteins. Amino acids and proteins are building blocks of life. When proteins are digested or broken down, amino acids are left. The human body uses amino acids to make proteins to help the body break down food. So proteins are polymers of amino acids, with each amino acid residue joined to its neighbor by a specific type of covalent bond. Is degradation of amino acids, and why does it occur? So let's suppose, I'm gonna explain this thoroughly. So let's suppose we have all the proteins that we want, and we have all the nucleotide bases that our cells can actually use. Yes, our cells can actually use. So what happens to any extra leftovers of amino acids? So well, you might think that it can be stored like fatty acids, which can be stored as triglycerides, or glucose as glycogen. So the big question is, can we store amino acids? The answer is a simple no, because our cells do not have the way to actually store extra excess of amino acids. So those amino acids must be broken down. So the majority of the breakdown of amino acids are in the liver. So let's see the definition of what I and my groupmates made. So this process usually occurs in the liver. So as I said, this process will occur in the liver. In amino acid degradation, the amino group is removed and turned into alpha keto acids. So in the process of degradation, the amino group is then removed and turned into alpha keto acids, which is then modified so that the carbon chain could enter the metabolism and actually become glucose or intermediates of the citric acid cycle. Do you know the alpha keto acids? So alpha keto acids are used primarily as energy for liver cells and in fatty acid synthesis, also in the liver. So amino acid degradation is a complex. If we say complex, we say it is hard. So it is complex web of reactions. It is necessary because unlike fats and carbohydrates, as I said, excess amino acids cannot be stored. The majority of the amino acid degradation occurs in the liver and skeletal muscle, as I said. So let's take a look at how we actually break down or catabolize the amino acids. Now we have the different ways to perform or the breakdown of amino acids, which are the transamination and the oxidative deamination, which is going to be reported by the next reporter.
Transamination leads to the same end result as deamination. The remaining acid will undergo either glycolysis or the TCA cycle to produce energy that the organism's body will use for various purposes.
Transamination is an exchange of functional group between any amino acid, except proline, lysine, and threonine, and any alpha keto acid. The amino group is usually transferred to the keto carbon atom of alpha ketoglutarate, converting the alpha keto acid to glycerine. Transamination reaction are catalyzed by specific transaminases, also called aminotransferases, which require pyridoxaphosphate as a coenzyme. So as you can see here on my back, we have the structure of amino acid and alpha keto acid, and so on and so forth. So amino acid have NH3+, and the alpha keto acid have double bond O. So ang ato aman kay transamination. So ang NH3+ kay i-transfer nato dinhi sa double bond O. So mahimo na ni siyang NH3+. So ang kaning O ma-transfer po siya dinhi. So mahimo na pud di siyang O. So ang pagkaresulta ang diba? Ang kani kay mahimo ng O. So ang as a result dinhi kay mahimo na siyang O. Plus diba dinhi kay NH3+. So dinhi gikan siya dinhi. So ang resulta niya kay naano siya dinhi. So ang matawag niya ani kay new alpha keto acid, and this is new amino acid. So dili sila mag-distribute kung dili pud sila di sila mag-distribute. So magkuan sila, mag-transfer silang duha. So mao na ni ang murag result niya. So ang gigamit nato sa pag-transferase kay mo ang transamination.
Another important example of a transamination reaction is the formation of aspartate, which is used during urea formation. In this case, the acceptor of the amino group is oxaloatate. For example, aspartate can be obtained from another amino acid such as alanine.
Transamination reaction catalyzed by specific transaminases are also called aminotransferases, which require a pyridoxal phosphate as a coenzyme. So there are different types of aminotransferases. The two major are alanine and aspartate aminotransferases. So here we have the alanine, which are our amino acid, and the alpha ketoglutarate, which is the carrier molecule that accepts the amino group from alanine. This glutamate pyruvate transaminase here catalyzes the movement of amino group from alanine to the alpha ketoglutarate. So if we transfer this amino group from alanine to the alpha ketoglutarate, we form pyruvate. And when the alpha ketoglutarate gains the amino group from alanine, we form glutamate. Next is the aspartate. Here we have the aspartate as an amino acid, and the alpha ketoglutarate, the carrier molecule that accepts the amino group from the aspartate. So if we transfer this amino group to alpha ketoglutarate, we transform oxaloacetate. And when the alpha ketoglutarate accepts this amino group from aspartate, we form glutamate. So this one, the glutamate oxaloacetate transaminase here, used to catalyze the movement of amino group from aspartate to alpha ketoglutarate that forms the oxaloacetate and glutamate. Now normally, the equilibrium constant for this reaction is about 1, so we can go this way or we can go in reverse. Another important example of transamination is the formation of aspartate. In this case, the acceptor of the amino group is the oxoacetate. For example, the aspartate can be obtained by another amino acid such as alanine.
Oxidative deamination is the first step to breaking down the amino acids so that they can be converted to sugars. Oxidative deamination occurs primarily on glutamic acid because glutamic acid was the end product of many transamination reactions.
Now that we know what oxidative deamination is all about, let us proceed to the process of the deamination. So the process begins by removing the amino group of the amino acids. The amino group becomes ammonium as it is lost and later undergoes the urea cycle to become urea in the liver. So when the amino group is removed from an amino acid, it is converted into ammonia through the urea cycle. The remaining atoms of the amino acid results in a keto acid, which is a carbon chain with one keton and one carboxylic acid group. The ammonia is then released into the bloodstream where it is transferred to the kidneys, which will secrete the urea as urine. So the urea cycle converts excess ammonia into urea in the mitochondria of liver cells. The urea forms, then enters the bloodstream, which then is filtered by the kidneys and is ultimately excreted in the urine, where the first reaction occurs in the matrix of the mitochondria. So what happens after it enters the bloodstream and filtered by the kidneys? So the filtered things, including urea, they pass out of the body through urine. As a matter of fact, on average, adults can excrete about 25 grams of urea through urine a day. As it goes stale, it returns into ammonia with the help of bacteria, giving the pungent smell of lavatories. And so the remaining portion of the amino acid becomes oxidized, resulting in an alpha keto acid. The alpha keto acid will then proceed into the TCA cycle in order to produce energy. This acid can also enter glycolysis, where it will be eventually converted into pyruvate. Now, pyruvate is the end product of glycolysis, which is converted into acetyl-CoA that enters the Krebs cycle when there is sufficient oxygen available, or it converts the original pyruvate molecules into ATP or usable energy for the organism. Now, TCA, or the tricarboxylic acid cycle, is also known as the Krebs or the citric acid cycle, which is the main source of energy for cells and an important part of aerobic respiration. Now, in the breakdown of amino acids for energy, the final acceptor of the alpha amino group is alpha ketoglutarate, which forms glutamate. Now, glutamate can then undergo oxidative deamination, in which it loses its amino group as an ammonium, which is the NH4+ ion, and is oxidized back to alpha ketoglutarate that is ready to accept another amino group, which can be seen in the picture. Also, in case you didn't know, which I know you did, but glutamate or glutamic acid is a non-essential amino acid found in a variety of foods, including both plant and animal-derived foods, such as bone broth, meats, mushrooms, and soy products. It is the most common form of glutamic acid in our bodies and considered a non-essential amino acid because of our bodies are able to synthesize it from other amino acids. This means we don't require this amino acid from our food sources, unlike with essential amino acids. Now back to the topic. This reaction occurs primarily in the liver mitochondria. Most of the NH4+ ion formed by oxidative deamination of glutamate is converted to urea and excreted in the urine in a series of reactions known as the urea cycle, which we already discussed earlier. Now, we all know NADH stands for nicotinamide adenine dinucleotide plus hydrogen. It occurs naturally in the body and plays a role in generating energy. The NADH produced by the body is involved in making energy in the body. Taking NADH supplements might affect blood pressure and have other effects. And lastly, NADH or nicotinamide adenine dinucleotide is a coenzyme that is a hydrogen carrier in metabolic reaction, especially in tissue respiration.
The breakdown of DNA and RNA is occurring continuously in the cell. Purine and pyrimidine nucleosides can either be degraded to waste products and excreted or can be salvaged as nucleotide components.
In the purine breakdown, the purine is composed of adenine and guanine. The purine breakdown is a uric acid, and uric acid is actually a toxic molecule, and we have to actually somehow get rid of it and excrete it properly. The daily production is estimated to be 600 milligrams per liter, and it has a low plasma solubility of 70 milligrams per liter. And so the uric acid enters the blood and it becomes sodium urate, and it also goes to your kidneys and gets excreted as urine. So this process is critical to get rid of uric acid from the blood. The purine breakdown actually starts with AMP and can get processed to adenosine. And AMP can also be processed to IMP as well by an enzyme known as AMP deaminase. And AMP can also be processed to IMP as well, and it is done by an enzyme named AMP deaminase. And as we can see here, there was an ammonium ion that is produced in this reaction. Adenosine can be processed to inosine by an enzyme known as adenosine deaminase, and again an ammonium ion is produced. And as we know, ammonium is something we need to get rid of, and that is done through the urea cycle. IMP can actually be reprocessed into inosine, and inosine can undergo another reaction and become hypoxanthine by the enzyme purine nucleoside phosphorylase. And by doing so, it releases a ribose 1-phosphate. So once we get the hypoxanthine, hypoxanthine can actually be salvaged in the salvage pathway, or it can further be metabolized into xanthine by the enzyme xanthine oxidase. And by doing so, it produces hydrogen peroxide. And xanthine can be further processed again by xanthine oxidase to uric acid, and again producing hydrogen peroxide. Xanthine oxidase is a critical enzyme where allopurinol, a drug commonly used for gout, is actually important, so allopurinol is an inhibitor of xanthine oxidase. So this means we don't have to metabolize hypoxanthine to uric acid. So it actually reduces your acid levels when you take allopurinol. Now, the other purine, GMP, can be processed to guanosine, which can then be processed into guanine via purine nucleoside phosphorylase. And again, it produces ribose 1-phosphate from the reaction. So once we get guanine, guanine can actually be salvaged by the purine salvage pathway as well, or it can be further metabolized by the enzyme guanase, while it releases another ammonium ion. And then it proceeds to xanthine again, which processes are still the same as mentioned. So GMP and AMP both lead to the production of uric acid. And this pathway is critical for the metabolism of purine.
Pyrimidine breakdown.
So in a pyrimidine breakdown, when DNA is degraded, you get a release of cytosine and thymine. Cytosine is actually processed through uracil throughout the amination reaction. Both uracil and thymine can actually be salvaged and repurposed in the pyrimidine salvage pathway. What happens to uracil and thymine is that they'll both be acted on an enzyme known as dehydropyrimidine dehydrogenase. The uracil becomes dihydrouracil, while the thymine becomes dihydrothymine. And this step is actually the rate-limiting step of a pyrimidine breakdown. The next step processes dihydrouracil into N-carbamoyl beta-alanine and dihydrothymine as N-carbamoyl beta-amino isobutyrate. And there's another common enzyme that processes these two products that is called the beta-uridopropanase. And what it does is that it processes the N-carbamoyl form of these two products into beta-alanine in the uracil pathway and beta-amino isobutyrate in the thymine pathway. And now these will actually be processed further. Beta amino acid butyrate will be processed into succinyl-CoA through a couple of steps. And beta alanine will actually be processed into acetyl-CoA. Both of these products will actually be used in the TCA cycle. And some of the byproducts include ammonia and CO2.
Purine breakdown.
Purine breakdown. The nucleotide will lose its phosphate through 5' nucleotidase. The nucleoside adenosine is then deaminated and hydrolyzed to form hypoxanthine via adenosine deaminase and nucleosidase, respectively. Hypoxanthine is then oxidized to form xanthine, and then uric acid through the action of xanthine oxidase. The other purine nucleoside, guanosine, is cleaved to form guanine. Guanine is then deaminated via guanine deaminase to form xanthine, which is then converted to uric acid. Oxygen is the final electrons acceptor in the degradation of both purines. Uric acid will be transported and dissolved in blood. Then the sodium urate crystal may appear in the acidic urines as small as brown needle-like crystal, which will then be removed by the body as urine. Most uric acid dissolves in blood and travels to the kidneys. From there, it passes out in urine if your body produces too much uric acid or does not remove enough of it. You can get sick. A high level of uric acid in the body blood is called hyperuricemia. Basis to uric acid. Purine catabolism means the breakdown of purine, because the end product of purine is the uric acid. Uric acid is produced when the body breaks down substances called purines. It is a waste product dissolved in the blood, then goes out of the body as urine. The common intermediate xanthine is where the adenine and guanine nucleotides merge. The enzyme xanthine oxidase converts hypoxanthine, which stands in for the original adenine, into xanthine. Deamination converts guanine to xanthine by releasing the amino group as ammonia. The majority of the ammonia will be transferred to the liver as glutamine and eventually excreted as urea if this process is taking place in tissue other than the liver.
Bases to uric acid. Both adenine and guanine nucleotides converge at the common intermediate, xanthine. Hypoxanthine, representing the original adenine, is oxidized to xanthine by the enzyme xanthine oxidase. Guanine is deaminated with the amino group released as ammonia to xanthine.
About an important of this purine nucleotide catabolism, it is the synthesis of uric acid and increase or decrease concentrational uric acid and related to their different messages. For example, gout, Lesnihan syndrome, severe combined immunodeficiency, purine nucleotide phosphorylase deficiency. So those are related to the uric acid. And coming to the degradation of purine, which is adenine and guanine. And these are the purine in the metabolism. AMP, adenosine monophosphate, IMP, inosine monophosphate, GMP, guanosine monophosphate, and last is nucleotide. And convert to adenosine to inosine and guanosine. Release the H2O inorganic phosphate by the exons of H2O nucleotides. And adenosine converted to inosine monophosphate formed by removal of amino group from AMP and adenosine respectively. AMP deaminase and adenosine deaminase. This is important deficiency like the severe combined immunodeficiency.
Pyrimidine catabolism. In pyrimidine catabolism, there are 8 steps. First, CMP and UMP and deoxo-IMP is converted into citidine, uridine dioxothymidine. This reaction catalyzed by enzyme nucleotidase. Step 2. Citidine is deaminated into uridine. This reaction is catalyzed by citidine deaminase. Step 3. Uridine and dioxothymidine is converted into uracil and thymidine. In this reaction is catalyzed by uridine phosphorylase. Here an inorganic phosphate is substituted on the first carbon of hydrolyzed glycosidic linkage sugar molecule. The sugar molecule is released in the form of ribose 1-phosphate and deoxyribose 1-phosphate. Step 4. Uracil and thymine is converted into dihydrouracil and dihydrothymine. This reaction is catalyzed into dihydrouracil and dihydrothymine. This reaction is catalyzed into dihydrouracil dehydrogenase. In this reaction, one NADPH positive H positive is oxidized into NADP positive. Step 5. Dihydrouracil and dihydrothymine is converted into B-uridopropanate and B-uridoisoborate. This reaction is catalyzed by hydropyrimidine hydratase. In this reaction, the cyclized molecule is converted into linear by cleaving the covalent bond in particular plate. Step 6. B-uridopropanate and B-uridoisoborate is converted into B-alanine and B-amino isoborate. This reaction is catalyzed by B-uridopropanase. The secondary product of this reaction is amino acid and carbon dioxide. Step 7. The B-alanine and the B-amino isoborate is converted into malonic semialdehyde and methyl malonic semialdehyde by transmissional process. This reaction is catalyzed by aminotransferase, and the secondary substrate is A-ketoglutarate, and the secondary product is glutamate. And lastly, step 8. Malonic semialdehyde and methyl malonic semialdehyde is converted into malonyl-CoA. These two products are entering the part of fatty acid catabolism like methyl-malonyl pathway. In conclusion, the pyrimidine catabolism pathway generally leads to NH4 positive production and thus diuresia synthesis. Thymine, for example, is degraded into methyl-malonyl semialdehyde, an intermediate of valine catabolism. It is further degraded to propanol CoA and methyl-malonyl CoA to succinyl CoA.